right, are we feeling ready for bios class on a Friday? So. All right, well, let's get into it. I feel good about this lecture, and I have a lot of checkpoints along the way. But first of all, I wanted to start with just a review of what we learned last Wednesday. So I want to give you time just to review some concepts, right? Take a couple minutes. Make sure you understand directionality. So I want you to label the directionality on this image. So label which side of each strand is 5' and 3'. And then I want you to write the complementary strand for this DNA sequence shown here. Right? So take a couple minutes and work through that. Yes. But you have to look at the molecules. Remember, when you label a carbon with it right side up, you start on the right hand side for this nucleotide. When you're labeling those carbons start at the right, you can do it. Talk about this together then. So if I start at this molecule in the top left corner here, what directionality would I label this? Would this be the 5' or the 3' end? 5', yeah, it's coming off of that fifth carbon here, right? So 1, 2, 3, 4, 5. Oh, I'm a little shaky because of these tiny little points here. But it's that 5' carbon, which would mean the other end, which would be 3', yes. So then on the opposite side of that 3', this would be the 5', the 3'. Yeah, very nice. All right, so 5' to 3', and the opposite strand is exactly the opposite, 5' to 3'. So if I were to write the sequence of that strand below here, right, I won't call on you to just say it here, but it would be the opposite, following those base pairing rules, A to T, G to C. All right, and hopefully you also wrote the directionality when you wrote the opposite too. So we know that this one starts with 5', so this one has to start with 3', because they're anti-parallel and opposite. All right, let's move on with a little bit more review. I kind of sped through this slide on Wednesday. So we talked about chromosomes and some chromosome terminology. All right, so chromosome is that discrete unit of genetic material. If we're talking about chromosomes that carry the same type of genetic material, they would be homologous pairs. Right? So maybe it's a chromosome from mom, chromosome from dad. They both carry the gene for eye color. The eye color might be blue versus green. Right? Slightly different, same type of information. Sister chromatids being identical copies of the chromosome. Right? So if we have our two homologous pairs here, when they replicate, right, they'll be joined in the center by this centromere here, but either side will be identical. Right? So we would call them sister chromatids. Right. So, good time to put this into practice with another labeling exercise. I'm only giving you a minute here because I feel like after just having talked about on the previous slide, and then you can flip back and forth, this shouldn't take too long. So what would number 1 represent? Homologous pairs, yeah. They're two different chromosomes, but they're the same type of genetic information. What about number 2? Centromeres. We're connecting the two sides of that chromosome together. And what would we call either side of a singular chromosome? Sister chromatids. Yes, very nice. All right. I think this activity is especially timely on a Friday when we all seem very tired and worn out. We're going to do a game, all right? And we are going to build strands of DNA and RNA. So I'm going to pass around piles of nucleotides. I want you to take the top 6. I think we can do 6 with the number of people we have here. I want you to take the top 6, and then with a partner or by yourself, figure out what type of nucleotide you have. All right, so do you have an RNA nucleotide or a DNA nucleotide? Right. Once you figure that out, you will bring them up to our cell here. Right, so the 2 tables are our cell. This is the nucleus of the cell. This is the cytosol of the cell. You'll place your nucleotides in a respective location. And then I'll need 2 volunteers to be our enzymes to make our nucleotides together via our sequence. Anyone want to volunteer to be an enzyme? All right. Yes. All right, we have Kendall and Isabella here. So let's see, you can be DNA polymerase. So you are going to link DNA nucleotides together. And you will be RNA polymerase. So you will link RNA nucleotides together. Right? So once you think you know what they are, come on up to the cell, right, and start figuring out how you might link these together. Now DNA will have 2 strands. RNA will only have 1 strand. We're making strands of nucleotides, DNA versus RNA. This exercise is really challenging you to make sure you know what RNA versus DNA is. Yeah, DNA because it's safe. Yeah. So if we're doing RNA, we can and so because of the enzyme, you can staple them together to start making a line. I just refilled it too, so that's all we're doing. I'm just making sure we're solidifying this information. We do that so we can get the new round with the blood pumping. So how are we differentiating between RNA and DNA? Yeah, sugars are a big difference here. So we had a mutation in the RNA. We had some DNA mixed in. Oh, wow. Look at this bag. Okay. Yeah, keep doing it. I was just talking with Angela. What could be the cause? Oh, you know what? That would be the highest release. Oh, mistakes? I don't remember this one. I'm looking at it. So, now to the point where I think it's fascinating. I want to challenge myself. That's my line. Yep. The RNA is using an enzyme molecule. The enzyme can line them up. Line them up and we can stop enzyme. You guys are coming. Hard to do. Yeah, go for it. Yeah, let's enzyme for as long as we're still sorting out our nucleotides. Yes, we are proofreading as we go. I'm solid enzyme. All right. I guess it. That's fine. It looks like they're all sorted, so if you want to stop them, stop. All right. Maybe we'll have to see about that. All right, so hopefully that got you up and moving, got you thinking a little bit. It's a good review. So how would you tell the difference between RNA and DNA? The sugar, yeah, the sugar. Ribose versus deoxyribose. Right? I like how as we were going along, if you saw a mistake, you were proofreading. Right? We'll see that happens in the cell. Right? Our enzymes can actually proofread. And when we put the DNA molecule together and see did our directionality and our base pairing, I love it. All right, so this is a precursor to what we will be learning today, because we're going to learn about DNA replication. All right, we're going to see how one molecule of DNA goes on to become two molecules of DNA, using our enzymes, those polymerase enzymes that we had with us, so first being one of them here. So if a cell divides, right, it has to replicate its DNA. Otherwise, if it divides, half of that cell, or that newly made cell, is not going to have DNA and it's going to be useless. Right? So we have to replicate our DNA when a cell divides. Right? Some terminology to be aware of as we're going through DNA replication is that the daughter strands are any strands that are newly made. Right? So these are the newly synthesized strands, whereas the parent strands are the original strands. Right? They provided the template for those daughter strands to be made. Right? And where does this occur? We nicely identified it in our table cells here, but where does DNA replication occur in the cell? Nucleus, because it occurs in the nucleus, right? So when we think about DNA replication and the history of discovering how DNA replicated, there was actually 3 different models that were proposed, right? All of these models took one DNA molecule and made 2 DNA molecules, right? But we didn't know quite exactly how this was done. And so one of the ways that this was proposed was this semi-conservative model, right? This is where one original DNA strand, right, would separate and create 2 DNA molecules, right? Each DNA molecule having a strand from the parent and a strand from the daughter. And you can see that with our 2 different colors here. The other method proposed was this conservative method, where we have our original molecule, and after it divided, one molecule was made up entirely of the original strands, and then one molecule was made up entirely of the daughter strands, or the newly synthesized strands. Right. And then there's this last method that was proposed, called this dispersive method. And this is where, after we replicated our original molecule, bits and pieces of both the daughter and the parent strands were kind of put together, and we can see that on both sides of our double helix on these daughter strands here, or these daughter molecules, we see bits from the parent, bits from the daughter, all kind of dispersed together. All right, so which model was correct? And I have this beautiful video. I absolutely love this video. I think it makes it really easy to understand.
So 2 scientists, Matthew Michelson and Franklin Stahl, had an idea to design an experiment that would be known as the most beautiful experiment in biology. Their findings are published in their article titled The Replication of DNA in E. coli, published in May 1958 in the Proceedings of National Academy of Sciences. These scientists used bacteria to answer the question of DNA replication, since it could be easily grown in the laboratory. They grew the cells in the presence of a specific type of nitrogen, which is found in DNA that would make all of the DNA very heavy. They used centrifugation, which can separate things according to their weight. Initially, all of the DNA in the cell was heavy and was at the bottom of the tube, since it was grown in heavy nitrogen. Then they started growing these cells in the presence of light nitrogen, so all of the DNA made in subsequent cell divisions would be lighter. After one cell division, the DNA was half as heavy, so half of the DNA molecule contained heavy nitrogen and the other half didn't. This is not in line with the conservative DNA replication model, which would predict that one molecule would be all light and the other all heavy. However, this is in line with the semi-conservative, or the dispersive model. After 2 cell divisions, the DNA molecule was now either half heavy and half light, or entirely light. So this is now not in line with the dispersive model, which would predict that the DNA after 2 cell divisions would contain a mixture of heavy and light DNA. Instead, this experiment agrees with the semi-conservative DNA replication model. Every cell gets one old DNA strand and one new one.
All right, so semi-conservative model it was through this very simple yet ingenious experiment done in the 50s. All right, so we know now that in DNA replication, right, when we have replication of the original double helix, one strand will be from the parent and one strand will be newly made or be the daughter strand. All right, so let's walk through DNA replication. So overview, big picture here, right? We have our original DNA, right, our double helix here. It's going to unwind and become 2 strands, right? Those 2 strands are the parent strands, and they will provide the template for the new strands to be made, right? They're great at being a template because, as we know, right, our nitrogenous bases have to follow the ATGC rule, right? Or an adenine is always going to bind to a thymine, and a guanine is always going to bind to a cytosine. Right? So if the template strand has an adenine, we know the daughter strand has to have that thymine. right? So it provides a really nice template to make that new strand. And the end result would be 2 new double helices with the same original sequence as our original strand of DNA, right? So taking one strand, splitting it apart, using it as a template, getting 2 new strands that are completely identical to the original. All right, but how is this done? All right, it's not as easy as what I just said here. And so we're going to walk through DNA replication knowing that this is a complex process. There's a lot that goes on, and we're going to touch on the key layers of DNA replication. Right? There are so many enzymes involved that we are not even going to barely scratch the surface. Right? But to start, right? The start of DNA replication, we start at the origin of replication. So this is a spot on a DNA strand that is where we're going to start DNA replication, right? This either has like some signaling proteins bound to this specific area. Sometimes it's a code within the strand itself. Lots of adenines and thymines sometimes will code that this is where we should start DNA replication, right? But it's this origin of replication, right? This is going to start to unwind and form a replication bubble, right? So you can see that replication bubble it looks like a bubble, right? It looks spherical in a way, right? And then at either end of that bubble, right, where it's starting to unwind on either end, right, this is where we have our replication forks, right? And this is where DNA replication is actively taking place. And as we keep unwinding our original DNA, right, DNA replication is going to proceed outward from the forks, right? So we have a replication fork on this end. DNA replication is going to go to the left, right? On the right side here, we have our replication fork. DNA replication is going to go to the right, right? So we are finding that spot in our DNA where we are starting replication. We are unwinding it in order for replication to proceed. But how exactly it winds is one of our first enzymes that we will talk about, is DNA helicase, right? So helicase is the enzyme responsible for breaking those hydrogen bonds between our nitrogenous bases in our double helix, right? So normally we have our hydrogen bonds holding our 2 strands together, right? DNA helicase is going to come along and break those bonds to separate out the molecule into 2 strands. Right? You can kind of think of it as a zipper that can only unzip. Right? It can't put it back together, but it can unzip our DNA. All right, so DNA helicase is coming along. It's unzipping our DNA. It's breaking those hydrogen bonds. Right? But once we break those hydrogen bonds and create 2 single strands, we don't want those strands to pair up again. Right? So to keep those strands separate, we have these single-stranded binding proteins. Right? So these are proteins that will bind to each of the separated strands to keep them open. Right? That way we don't immediately just reform a double helix. All right, so helicase is unzipping the DNA. These single-stranded binding proteins are binding to those strands so it doesn't rezip itself together. All right, but we have one more enzyme playing a role in unwinding our DNA. And this is the DNA topoisomerase. Right? So this enzyme relieves any additional coiling that might occur. So I don't know if you've ever peeled apart a piece of string, right? If you peel it apart really fast, sometimes it can get super coiled. Where it needs to peel apart, it can get knotted, it can get messy, right? Same thing can happen here, right? If we're unwinding our DNA, our double helix that is starting to unwind can get super coiled. Right? So this enzyme plays a role in preventing that supercoiling. Right? What it actually does is pretty cool. Right? It actually completely cuts apart the DNA here, unwinds it, and puts the DNA back together. Right? That way helicase can do its job. That way we can start with our DNA replication. All right, we're going to step back. I want you to answer so far what has happened to our DNA strand and what have been those proteins used, and as a bonus, what are their functions. And I'll give you a few minutes to do so here. Makes it easier when I come out. All right, so what has happened so far in our DNA knowledge? Yeah. Yeah, we have started to unwind our DNA. Where does it start to unwind at? Origin of replication is what starts to unwind, right? That forms what? Forms the replication bubble, which at either end of that bubble is going to keep unwinding at, what are those 2 points? The forks, yes, the replication forks. All right, so DNA at this point has unwound. All right, what proteins have we used? We've used helicase. What does helicase do? Unzips, yes. What is our other proteins that we might have used? Yeah, those single-stranded binding proteins. We got to keep those strands apart. And our last enzyme that we're using. I always have to slow down when I say this word too. Topoisomerase. Yes, it's preventing that DNA ahead of unwinding from getting too coiled and tangled. Yes. All right. So we have unwound our DNA, but now we have to actually build those newly built daughter strands, which involves a lot more proteins and enzymes. And so we'll walk through this step by step in the same manner here. All right, so after these strands have unwound, right, an enzyme is going to come in called primase. Right? This enzyme is going to put an RNA primer on one of our template, or on both of our template strands. Right? So it has this primer sequence of RNA molecules. Right? So match that sequence up to a portion of our template strand. Right? So it's putting RNA molecules in place. These strands or these sequences are only about 5 to 10 nucleotides long. They're not very long, but the purpose of this primer is to signal where to start creating our new strand. Right? Now, because this is RNA and not DNA, eventually we'll see that this RNA primer is going to be replaced with DNA nucleotides. Right? But we have to provide this starting point, and primase is putting this starting point in place so that our next molecule, DNA polymerase 3, can recognize where to start and build these new strands, right? So DNA polymerase III is responsible for building the new strands. It's taking these free-floating nucleotides, covalently bonding them together into those phosphodiester bonds to create the daughter strand. Right? So it requires this RNA primer to know where to start DNA synthesis. Right? And this enzyme can only work in the 5' to 3' direction. Right? And we'll see some complications that arise from this in a couple of slides here. All right, so we have our primase, put on our primer. Our DNA polymerase recognizes that primer, binds to that area of our DNA molecule, and starts to build that daughter strand by putting in the new nucleotides. Right? Now it knows which nucleotides to put in because of our base pairing rule. Right? So if our template strand has that adenine, it knows to put in a prime. All right, so we have two strands, right? Because we have unwound a double helix. So this first strand, we call it the leading strand, right? That template parent strand. On one end is our 3', and the end that is constantly unzipping from helicase is our 5' end, right? And we know that strands are anti-parallel, so when DNA polymerase comes along and starts adding in those new nucleotides, it can only work in that 5' to 3' direction, but for this leading strand, no problem. Right? It's going to find our RNA primer, and if you can notice it here, it's slightly darker in color on this image. It's kind of subtle, right? And this primer, when it makes the new strand, is going to go in the 5' to 3' direction. Great, awesome, all right? It's going to work continuously. As helicase keeps unzipping that DNA, we're just going to keep on adding more nucleotides and creating this really long, continuous strand, all right? But that doesn't work for the other side, which is why we call the other strand the lagging strand. Right? So if we look at the template strand here, we see it's unzipping at the 3' end, right? And then going towards the 5' end. Right? Well, our polymerase can only work 5 to 3', right? So it has to start at an RNA primer and work 5 to 3, and it goes away from where the helicase is unzipping our DNA. Right? So essentially it has to do this in fragments, in segments. It can't just keep on following that helicase along because it's the wrong direction. So what will happen is we'll have a primer, right? The primase adds that primer. Our DNA polymerase III comes along and builds a section of our new daughter strand, and then it will stop when it hits the section it has already built, right? And then it's going to come along, find that primer, build another section until it hits that section that it has already done. And as this unzips, it's going to continue to make these little fragments, right? We call these Okazaki fragments after the researcher that discovered this, right? These fragments, they're not long and continuous. They can't be, right? Because DNA polymerase does not work in that direction. And they tend to only be like 100 to 200 nucleotides long. Right? So we're building these fragments. Do we have a question?
So does it start with a little bit that is the right one, the one that Paris kind of
Yes.
moves to the left and then goes right again?
Yes, because if you think at one point this is where it was unzipping, so this is the part of the lagging strand that was free to build the first fragment. And then we unzipped more, and so we could build another fragment. Unzipped more, we could build another fragment.
So the one that's right goes to one strand in the opposite direction of the
Opposite direction of this arrow, yes. It's building it in the direction of these dark blue arrows.
but it just starts on the right hand
Yes. It starts on that new section of that strand that has been newly unzipped. Right? And it can't go all the way because eventually it's going to run into a section that it's already built. Right? I'll have a visual at the end of this that'll help kind of put this together. All right, so we're building our sections. We have that leading strand. It's built one long, continuous segment, right? That lagging strand has built all these different Okazaki fragments, right? We know that that RNA primer put in RNA nucleotides So we got to swap out those nucleotides with DNA nucleotides. And that is the job of DNA polymerase 1. Right? It's swapping out those nucleotides, it's removing the RNA and putting in the DNA. Right? Now, RNA polymerase 1 can swap out those two different kinds of nucleotides, but it can't join the different fragments together. Right? So it replaces the nucleotides, but it can't join those Okazaki fragments together. And that's where we have our last protein that we will talk about here, called ligase. Right? Ligase is then going to come through on both sides here. It's going to put these segments together. Now, it really only works on the lagging side because that is where we have our fragments. But if any fragments were to occur on the leading strand, it would also glue those back together. Right? So ligase is connecting those Okazaki fragments together. All right, well, let's watch a visual that puts all of this together, and hopefully it helps connect the dots in your mind here.
Cated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both
DNA
strands of
polymerase
DNA.
1. Old terminology.
Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand.
All right, so hopefully this helps kind of put that image in your mind. Yeah, what questions do you have?
What does the DNA
Yeah, so if it reads with an A, right, a DNA nucleotide would be a T, but if it's RNA, it would put a uracil in that place, which is why we need to go back and swap out those nucleotides to make it all of it DNA. Yeah, good question. A new enzyme? I don't know for sure, but I would assume there's just multiples of these enzymes continuously working. Yeah, at least two working, if not more, on that lagging strand. Yeah.
starting
Yes.
5 to 3, and in this direction, this is 5 over here, but that's not in this direction, we're going to 5.
Right. So, yes, it depends on how you look at it and where you're starting and what orientation. You just always want to pay attention to the leading or the parent strands and what direction that parent strand is in. 5 to 3 or 3 to 5. That makes sense.
Yeah, but like, as the whole process is happening,
Mhm.
you just have like a leading or lagging, same strands throughout the sides.
You mean in that replication fork? In that bubble?
Yeah, these are 2 replication forms.
Mhm.
So, starting from the left to the right,
Mhm.
that leading, and then the same thing sliding on the direction. So you just have 2 of each on both sides.
So you only have one of each, right? But one's going to be unwinding. So that first, this is a great question. I never thought about it like this. So as we're unwinding the DNA, right, we have those 2 replication forks. One's going to be unwinding in a lot of the examples that we've talked about so far, right? The other is going to be unwinding in that opposite direction, which I think what we're getting at here is that lagging strand, as it's unwinding to the right, it kind of becomes a long continuous piece because it doesn't have to stop because it's keeping unwinding, right? I've never thought about it. I've never looked at it. I would assume that we would have a really long continuous piece until that DNA molecule ends on that side. And so that Okazaki fragment would be a fragment, but a really, really long fragment, is what I would assume what would happen. And then eventually, we're going to run out of stuff to unwind. It's going to stop. But we're going to, on the opposite side, keep having to make those little fragments ahead of that long one that it just made. I would assume that's how it works. That would make sense to me in how it would work. That's a great question. I've never looked into it. Yeah. Do you have any other questions? All right. I want to get through, we can definitely get through one more slide here. So DNA replication, right? There's a lot going on, but it's actually really accurate, right? There's ways that DNA can make sure it's accurate. So hydrogen bonding between those nitrogenous bases, right? We know that between A and T, there's only 2 hydrogen bonds, and between G and C, there's 3. And so if there's a mismatch in how they're binding together, those hydrogen bonds are just not going to be as stable, right? So when we bind these nitrogenous bases together, we really want to keep it stable, right? We also have this DNA polymerase molecule that is able to proofread, right? It's really unlikely to form bonds if the pairs are mismatched, again, because those hydrogen bonds would be unstable. But it can also remove any mismatched pairs that it might have made. Yeah, Max.
What happens to our RNA when it's activated
I would assume that we use, because biology is all about efficiency, if they don't need to be used. I don't know what would happen. I don't know if they'd be recycled by the cell and made into other types of molecules, or if they just don't turn out. No. All right, so very accurate. We have these means of proofreading. All right. We have time to go through this review here. So let's end with our review of DNA replication itself. So we already talked about the proteins that helped unwind our DNA strands. Now we're looking at DNA replication. Right? So I'll review what direction is DNA replicated. Right? How is replication different in the leading versus the lagging strand? And what are those 4 proteins used in building this new DNA strand? Does it make sense? DNA was what your question was? I think it would. So that's a lot of videos. I have a lot of supplemental videos attached at the end of this slide. Visuals, that's how I learn. I do a lot of visuals. Some knows right. It might be like environmental pressure, signals that we can see. It's a whole host of reasons why this cell might start to replicate. But assuming that cell knows that it's replicate, there's no we'll get to my pictures and the cells I'll go on Monday. Ooh. It's not the concept of hours. I'm trying to think how long. I want to say it's a couple hours. But that would be I just read it in my mind. It is they had a very fast. Okay. All right, so in which direction is DNA replicated? 5' to 3', yes. How is replication different in the leading versus the lagging strand? Yeah, leading is one continuous molecule versus the lagging. Yes, lots of Okazaki fragments. And what are the proteins that we are using in replicating our DNA and building that new strand? What's the one that adds on the primer? Primase. Yes, what's the one that is adding in the new nucleotides? Polymerase 3. What's the one that's replacing the RNA molecules with the DNA molecules? Polymerase 1. And then gluing everything together, we have an enzyme? Ligase. Yes. Now notice there's no DNA polymerase 2. All right, DNA polymerase 2 is still kind of an unknown variable. There's a lot of research going on it and what it does. We think it's a backup polymerase and that it can proofread. We don't really know yet. All right, so homework for Monday, if you have that content check due and those exam corrections due, and then just upcoming next Friday, there's actually no class because it's a faculty work day, so keep that in mind. All right, enjoy your weekend. Hopefully you have some fun Halloween stuff planned.